So yeah, I was assigned the task of actually talking about tackling undruggable proteins. And before we get started, I just want to make sure that everyone understands we're not going to be talking about making drugs tonight. We're going to be talking about small molecule discovery aimed at difficult targets. Before we get started, I actually have a couple of business items. So when I give lectures, I like to move around a lot and sort of do a dance. And having been an audience member in this auditorium in the past, I know that, especially for you folks sitting in the front row, it can be difficult if I move away too much from the podium. So I'm going to try to stay at the podium. If I get in your way, let me know, and I'll scurry back. Another business item. So I think, as many of you know, it's sort of a cardinal rule every time you give a talk that you should know your audience. And I've been sitting in the last couple of seminars during this series, and I have to confess, I totally don't know this audience. So being a scientist, I figured we would make a measurement of the audience before the talk actually started. So if you're a high school student, raise your hand. All right, good. Good. If you're a parent of a high school student, raise your hand. Excellent. If you're a high school teacher, raise your hand. Excellent. How about the college students and the undergraduates? Excellent. Okay. Okay. What about the graduate students and postdocs? Okay. What about the people who identify themselves as everyday scientists working in the lab? What about the people who are not everyday scientists but think science is just totally awesome? That's awesome. Great. What about the people who work here at the Broad Institute and the people who do not work at the Broad Institute? Excellent. Excellent. I guess the final question, since we're going to be talking about both chemistry and biology today, how many of you would identify yourself as chemists and biologists? Wow, more bio people. That's cool. I'm actually, my PhD is in chemistry, so this will be a little different. OK, all right, so let's get started. I think since there's so many students, one thing I should say is that a sort of a key defining moment that differentiates my time as a student versus your time as a student is the sequencing, the completion of the sequence of the human genome. So when I was a student, we had already seen that there were a number of sequences completed for model organisms such as rice and corn, worm, fruit flies. But we did not have the human genome sequence completed. And so seeing that organisms such as rice and corn had 45,000 or 50,000 different genes, we just assumed that humans would have something like 100,000, 150,000 genes. Right around the time where I was getting ready to finish my PhD and move to the Broad Institute, the sort of draft sequence of the human genome was completed due to the efforts of many, many different people, but quite a few people here at the Broad Institute. And it was a bit of a shock when you find out that, in fact, we have 21,000 genes. And don't be offended, right? It's not that you're somehow less complex than rice or corn. Maybe some people are, I don't know. So this actually presents a really cool, interesting challenge in biology, sort of in this post-genome era, and that is, if the total number of genes does not give rise to organismal complexity, what does? And so in the first two seminars, part of this series, you heard both Mark Daly and John Rinn talk about some of the other things that might give rise to organismal complexity. So we like to think about proteins, and so we think, some people might think 21,000 genes encodes 21,000 proteins. In fact, it actually encodes many, many more proteins through processes such as alternative splicing and post-translational processing, where you might stick a phospho group or a sugar onto that protein. So we don't really have 21,000 parts. We actually have many, many more parts. And when you just think about proteins, we may have something on the order of 100,000 different proteins. Those proteins are mostly of unknown structure and unknown function. And so it's a challenge to try to understand the roles of those proteins. This is the sort of thing that we think about here at the Broad. And we've assembled, biologists, chemists, mathematicians, physicists to think about these types of issues. All right, so this is a slide that I blatantly stole from David Altshuler, and I really like it. It's essentially showing you the different types of genes that have been associated with disease, starting around the time that the human genome sequence was completed. And so we've color-coded different types of diseases here with the genes. You can see somewhere around 2000, this gene PPAR gamma, which is a transcription factor, becomes associated with type 2 diabetes. We kind of plug along each year and identify a new gene that's associated with disease. And then right around 2005, 2006, we start to see an uptick in the number of genes that are associated. And 2007, I'm actually only showing you the first 9 months of 2007, because if I were to show you all of 2007, we'd actually go all the way up to the top, beyond the ceiling of this auditorium, and I'd need to borrow Al Gore's cherry picker to actually go to the top. And of course, we can't even show 2008 and 2009. And the last time I saw David speak, he said that there were more than 500 genes that were associated with disease just during this era. So what does this mean? We're seeing sort of a tidal wave of new types of targets. And when you go and you ask what are these particular genes, some of them are what we call the usual suspects. These are the types of genes that we know are associated with disease, things like certain types of kinases, certain types of proteases. But actually, a lot of these targets are things that we've never seen before. We just don't know what they do. Or we know what they do, but they're sort of more difficult to study. So this presents a challenge for scientists here at the Broad. And as you've heard in some of the previous seminars, we, as an institute, have developed sort of complementary approaches to studying function that are rooted fundamentally in genetics. We also have a very strong group here using RNAi interference to try and understand the role of different genes and proteins. I'm part of the chemical biology group here, and so we spend a lot of our time thinking about how small molecules can be used as tools to directly at the protein level. So we would like to be able to have compounds that bind to proteins and that either activate or inactivate their functions. And we think those compounds can tell us quite a bit about the phenotypic consequence of modulating that protein. Why do we like small molecules? Well, we've got a list here. Sometimes they can be used reversibly. So if you run an experiment and you run it with a small molecule, you can sometimes remove that small molecule and sort of rescue or recover an effect. They can be used under various conditions. So you may have a protein that binds to calcium, and it has one shape when it's bound to calcium. But when it's not bound to calcium, it has a completely different shape. And you may be interested in looking at experiments under both of those conditions. We can administer different doses. I think importantly for me, I think that small molecules are important tools because they allow you to have temporal control over an experiment. So if you think about knocking out a gene from an organism, sometimes that gene is critical and the organism will die. We have a chance to actually knock down the function of that protein with a small molecule and not necessarily kill that organism. For non-essential genes, the organism may have a chance to reach something called a steady state, so it can overcome losing that gene. Whereas here, with a small molecule, we can understand the process of losing that function in time. We can also use small molecules with spatial control, and sort of importantly, we can think of small molecules potentially as therapeutic leads. So what is an undruggable protein? I'm kind of curious to know what this means to this audience. So I was wondering if there might be a couple of volunteers who would actually give their version of what an undruggable protein is. Anyone? Because if you don't answer, poor Derek Martin in the audience is going to have to actually give an answer. Derek.
Something that's therapeutic nature has been developed for scientific and very challenging.
Okay. Anyone else?
We did not do that.
So Derek was saying that it's a protein target for which it's been difficult therapeutically to target that particular protein. Anyone else? Yeah, I see a hand here. Yeah, that's good. Highly complex protein. Anyone else? Can't drug it. Okay, that's the obvious one. So this is all good. So I think one thing about that, the reason why I didn't like the title of this talk is because it uses this word undruggable, and undruggable means totally different things to totally different people. To some people, it means this is the part of the proteome that we've never been able to, attack with a drug. For some people, it means what Derek said, that is, it's a very difficult, challenging target. And for some people, it means that it's a part of the proteome that we'll never be able to drug. So I'm just going to kind of go through what we think here at the Broad. One sort of piece of information that's interesting is the number of molecular entities targeted by the full sort of pharmacopoeia on the market is 482. And I just told you that there are 20,000 genes. So that's a paltry number, right? And actually, there are people who say this is not even the correct number, that when you correct for redundancies and targets that really shouldn't have been targeted to begin with, that maybe it wasn't the right target, maybe it was a mutation, this number actually drops to closer to 300. So we're not historically doing a very good job of looking at many, many different targets. There may be many reasons for that. Examples of the things that have worked are things like G protein-coupled receptors, kinases, proteases. So as the pharmaceutical industry and other groups, including academic groups, have identified small molecule matter to these types of targets, we then go back into our labs and synthesize additional compounds, design compounds that actually target these types of targets. Those types of compounds go into our screening collections, and then they're screened against new types of targets. It's kind of a self-fulfilling prophecy in a sort. But these are the guys that we call the usual suspects. As someone said, the proteins produced by the majority of genes are just considered undruggable. And that may be because they're very, very difficult. It may be because we just don't have the tools or the compounds. Here at the Broad, we like to make sure that people know that when we say undruggable, we are referring to that specific molecular challenge that Derek referred to, that really it's not that the compound or that the protein cannot be drugged, it's that we haven't come up with the right chemical matter yet. And so because of that, we like to not call undruggable targets or undruggable proteins undruggable. We like to use other terms such as difficult. Often when you talk to people, they will call them obstinate. And my favorite is recalcitrant. And these types of targets tend to be things like transcription factors, things like proteins that interact primarily via protein-protein interactions, as opposed to some of these proteins which function primarily by converting. They do chemistry. They convert one thing into another thing through some sort of chemical transformation. And as a chemist, I can probably grab some of the chemistry students here and have them go to the board and actually draw you structures of things that would target these types of targets. But as a chemist, I cannot tell you that I know any rules that will allow me to target transcription factors or protein-protein interactions. We just haven't developed that rule set yet. So we like to think quite a bit about small molecule screening here. And at the Broad, we sort of think there are two key items to sort of, moving beyond the lamp post. We'd like to sort of search beyond both the protein space and the chemistry space that's been searched to date. And so we think the first part of that is to assemble a transformative collection of small molecules for screening. That means a lot of different things. Here at the Broad, we try to build a set of compounds that are very diverse, include things such as natural products, include compounds that are synthetic and may have been designed to have certain types of physical properties. This is actually the topic of a seminar on its own. And so I'm not really going to discuss this. I'd recommend to Megan that maybe next year she invite one of the chemists from the chemical biology program to talk specifically about this issue. I'm going to focus more on developing new screening technologies, and there are many people here at the Broad Institute and other institutions and industry who are thinking about this particular challenge. And I'm just kind of curious, how many of you from John Rind's talk actually remember his magic glass slides? Okay. What we do actually involves magic glass slides too, but before we get into the details of what my group does, I just want to kind of acknowledge that the larger chemical biology team really takes more than one approach. And when we think about Undruggable, we not only think about proteins, but we also think about processes. And there are individuals, many individuals in the chemical biology program who are thinking about what we call state switching screens. So they have a specific phenotype that they're interested in, and they screen these different small molecules, and they look for their pinch of magic dust that induces some change in phenotype. And then the challenge is to actually try and take that pinch of magic dust and identify the protein in cells that it binds to and modulates. This is really a challenge, and there are a number of people at the Broad Institute who are trying to come up with systematic approaches to going from pinch of magic dust to a cellular target, and that would be the proteomics group here at the Broad. My group spends most of our time sort of thinking about that tidal wave of targets that I showed you on Dave Altshuler's slide. And what we do is we basically pick our favorite proteins, and what we want to find are compounds that directly interact with that target of interest. And then the next step for us is to go into cells and try and understand what are the phenotypic consequences of binding to that protein and modulating it in cells. So what we do is we then take our pinch of magic dust and put it through a broad swath of different biological studies. Is that clear to everyone? It's sort of, we think of them as complementary approaches. And I'm not going to go into all the details on this slide, but if you're thinking about binding, I mean, we think binding, high throughput, general binding assays is one way to try and quickly go through and identify compounds that may activate or inactivate proteins of unknown structure or function. That's the challenge. We can't design these things, so we're going to screen for them. And there are historically a number of really great ways to think about ligand discovery, small molecule discovery. And I've just put a couple of examples up here. One of my favorites is isothermal titration calorimetry. I'm not going to go into the details, but for the students here, I'd suggest maybe writing some of these terms down. Do a Google search. I guarantee you this is going to be a Wikipedia hit. The people who work in my group over the summer have these notebooks, and they basically have the Wikipedia lists during meetings. So ITC is great. We love it and because it gives you high-value thermodynamic information about the binding interaction. But it's not really high throughput enough to let you go through the entire proteome. And it requires quite a bit of sample. So we don't really think of it as a way to screen many, many different proteins against many, many different types of compounds. And we tend to use this sort of as a follow-up approach. Fluorescence polarization is another great assay that I like to use. It's essentially when a protein, an entity will rotate in solution at a certain rate. And as you add to that entity and make it larger, it has a change in rotation in solution. And so when something binds, you can measure that change in rotation. And this is a great assay because it allows you to look for binding in high throughput formats. So we have these little plates that we run these assays in. And we can look at many, many different compounds against a single protein. But the problem is to set this assay up most intelligently, you need to understand something about that protein in advance. And you often either need to label the protein in some way or put a fluorescent label on the compounds to measure that rotation in solution. Another approach that we've used in the past is called the on-bead binding assay. So the idea here is that you've got a collection of molecules. Maybe it's 2 million different molecules. And they're linked to some sort of solid support, some small bead, like really teeny little beads. They're 90 micron in diameter. Each bead has a different compound. And what you do is you take your favorite protein, this orange thing here, it's got some sort of fluorescent thing on it, and you incubate it with that set of beads. And basically what you're looking for are compounds that become fluorescent. Anywhere you see a fluorescent compound, you have a potential interaction with that small molecule. This is a great assay that will allow you to look through a very, very large number of compounds. But the problem is it's really very limited in the protein dimension. So you can come in with your favorite protein. You have to sort of wash the beads to come in with your second favorite protein. You have to wash the beads to come in with your third favorite protein. And each time you subject the compounds to potential damage. So we wanted to come up with something that was a little bit like the on-bead binding assay, but that would allow us to sort of dramatically expand the protein dimension. There's some other great methods here, but what we've done is to take our cue from the cDNA microarray world and make those magic glass slides that John Rinn was talking to you about, except our magic glass slides actually have compounds on them. So what we do is we start with these 384 well plates. Each plate's got a little divot, 384 of them. Each well has a different type of small molecule. And then each molecule is in a solution called DMSO. And we use robots to actually pick up the solutions. We pick up about 200 nanoliters of solution. And then the array will robotically print about a nanoliter of stock solution onto a series of chemically derivatized microscope slides. We usually do 100 of these at a time. That 1 nanoliter will result in a spot that's anywhere between 150 and 300 micron. We can put 10 to 12,000 different spots on a single glass microscope slide. And then what we do is we incubate these slides with our solutions of favorite protein. And just like those beads, anytime a protein hits a spot where it likes the compound, we get a fluorescence signal. So like I said, we can manufacture 100 of these replicate slides at a time. And because we use so little compound, we can make enough slides to actually think about screening the entire proteome. And this is sort of a schematic of what the assay looks like. This is your slide. We've got compounds being displayed. We come in with a protein that binds. Usually these proteins have some kind of tag on them. And then the fluorescence comes from an antibody that binds to this epitope tag. We can do this assay with purified proteins, but in recent years, we've actually started to also incubate the slides with solutions of cellular lysates, and we detect binding of proteins directly out of those complex mixtures. And this is what a real piece of data looks like. This is a slide that has 10,800 different spots, and we've incubated it with protein, and you can see there's some red spots, and this is a zoom-in of spots. So presumably there's a protein binding to a small molecule in each of those spots. And we have sort of developed a pipeline for evaluating these compounds. So every time we screen our favorite protein, we screen on at least 3 replicate slides. And we convert the fluorescence intensities on all of these spots into some sort of screening metric. We tend to use something called a composite z-score. And what we do is we look for compounds. This is a distribution. So red here is a spot where we didn't put any compound. Blue here is a spot where there's a compound. And cyan, these high signal outliers, are the things that light up. So these are the things that we would call a hit in our assay. We also look at all of the different replicates, the 3 different replicates, and what we try to find are compounds that fall along a perfect line of reproducibility amongst the 3 different axes. And these are often the things that we would like to follow up in additional biology assays. So I mentioned, we'd like to not just look at one protein, but we'd like to look at many different proteins. And in fact, our group has now looked at close to 300 different proteins. And in doing so, we're in a position to make specificity heat maps or other types of analyses that allow us to understand, does this compound bind to one protein? Does it bind to a bunch of related proteins? Does it bind to no proteins or every single protein that sees the glass slide? So we use a combination of this type of data and this type of data to follow up or to prioritize candidates for follow-up into different types of phenotypic or functional assays, as well as secondary binding assays. So we use a technique called surface plasmon resonance. It's another Wikipedia word. SPR is just another binding assay. And what we see is that 80% of the things that we call hits here will retest in one of these secondary binding assays. And they usually have a range of affinities from low nanomolar, which is a really good affinity, to 50 micromolar, which is not so strong. All right, so I mentioned that we have chemically derivatized glass microscope slides, and we've spent probably 10, 11 years working on different types of surface chemistry, and I'm not going to bore you with that, especially because most of the hands said that you were biologists. But what I will say is that it's actually really, really important, and we've probably worked on about 30 different surface chemistries in total over the last 10 years. I'm just going to present the two that we really use today here at the Broad Institute. They're sort of our bread and butter chemistries, and we use them for different purposes. The first one's got a kind of a cool title, homogeneous capture via fluorophilic interactions. So are you guys all familiar with Teflon? Why is Teflon cool? Nothing sticks to it, right? So when you're working with surfaces and you're working with proteins, you're always worried that the proteins might kind of glom up on that surface and stick non-specifically. We call it adsorption. It turns out that we're using an attachment method here that's sort of, it resembles Teflon. So we like it because we can basically take compounds that have what we call a fluorous tag. It's basically a bunch of carbons linked to a bunch of fluorines. And then we can make glass surfaces that have the same type of coating on them. And it's kind of like something called the hydrophobic effect, where grease likes grease but doesn't like water. Here, fluorine likes fluorine but doesn't like water. So we can just put the slides into water, and the spots will stay nice and tidy. We use this chemistry in specific cases where the orientation of the molecule is important. So certain types of molecules, like this compound here called varinostat, which is actually a clinical agent, have special parts on the molecule that are required for binding to the protein. So this is called a hydroxamic acid. This thing is actually a chelator group, which is required for interacting with its target, which is called the histone deacetylase. So if you were thinking about putting this on an array, you'd need to think about the orientation of that compound. The orientation is very important. We want this thing, this chelator group, to be face up on the array. So here, we have many, many people interested in studying the target of these particular compounds. And what they did was they synthesized a library of these compounds, and each of these compounds had that little fluorous tag appended to them. And then what we did is we used our robotic arrayer to print these types of compounds onto the fluorous slides, and the compounds are oriented face up. And so using this approach, we were actually able to identify new compounds that bound to these histone deacetylase targets. And some of the structures of the targets are shown here. So this is one of the methods we use when orientation is important. But for most of the experiments that we do here, we're just running a screen. We don't care. We don't care if the orientation is right or wrong, because we're looking at targets for which there is no known chemical matter that binds to them. We're just looking for anything that binds. And so what we'd like to do is have access to as many, many different types of compounds as possible on a single glass slide. And that's a challenge because, as a chemist, it's nice to think about this specific reaction, or the specific functional group will react with the slide in a specific way. But in order to maximize the number of compounds that you can screen on a single slide, we needed to take a step back and not think about chemoselective reactions, but actually promiscuous reactions. And so our solution to that was actually to make isocyanate-coated surfaces, because many different functional groups that you see in compounds from nature, known drugs, or synthetic sources, actually have reactive groups that will react with an isocyanate. I won't go into the details, but we basically put a surface that's got a terminal isocyanate on the arrayer. We print our compound collections, and then they react with the slides. And then we add a catalyst that essentially boosts the reactivity of certain sluggish functional groups. And this is what a printed slide would look like. What we've seen is that probably somewhere between 70 and 80% of commercially available screening collections are compatible with this specific chemistry. And so the take-home message is that you can make slides that look like this. So you can make slides that have compounds from a variety of different sources. In the outer ring, these are structures for compounds that are natural products, known bioactives, FDA-approved drugs. And in the inner ring are structures, core scaffolds for some products of diversity-oriented synthesis or combinatorial chemistry. And what I've done is color-coded the different groups on these molecules that can actually attach to the surface. And as you can see, many of these molecules have more than one functional group on them that will sit on the surface. And again, like I said, as a chemist, you want to know exactly how it's oriented. But what we found is that this is an advantage. Because in a single spot, when you come in with your favorite protein, that protein can actually sample multiple modes of binding within a single spot. And if we're just trying to find any kind of interaction, this basically increases our power to identify things. We've also used this chemistry to print natural products as well. This is just a slide to show you some of the representative interactions that we've discovered using this approach. Some of them are druggable targets and some of them are undruggable targets. But it's a very diverse set of structures, very diverse types of proteins. And this has really been a community effort. So what we see here, this is a list of chemists from not only the Boston area but around the world who have compounds in their labs. Maybe they're products of a total synthesis. Maybe they're products of a combinatorial synthesis. And they send them to us at the Broad, and we put them on our arrays. And this is a list of some of the folks that we've interacted with that have their favorite protein that they want to put on the arrays. And the names in blue are the names of our collaborators where we actually found something. We found a hit on the array. We found binders. And then the names in red are the names of investigators that have not only binders, but compounds that already have demonstrated function, have been published, or for which intellectual property has been generated. So all of that data with all of those investigators you just saw, and we generate a lot of screening data, not just with the microarrays, but with these cell-based assays, and we need a place to put it. And so what we decided was we were going to put all of this data into the public domain. Usually after a year of running the screen, we promote the data to a tool called ChemBank. This is the URL. It's meant to be a tool for the community. So if you have your favorite compound and you want to see if that compound has done anything in any of these assays, you can go to ChemBank to see what it's done. If you have your favorite protein and you want to see if anyone has a compound that binds to it, or if there's a compound that binds to it but also targets some sort of cellular function, you can use ChemBank to do that. And our sort of key applications of ChemBank are, again, trying to make connections between, say, a small molecule microarray-based binding assay and ask what that compound did in a very large swath of functional assays. We also use this as a tool to try and understand the relationships of different structures in the screening collection to activity. And I'm going to just quickly kind of go through a couple of vignettes that involve proteins that have been screened that are classified as undruggable. So the first target is actually called sonic hedgehog. It's a target that's very important in development and also in several types of diseases, especially cancer. And the hedgehog gene was originally identified in a large-scale genetic screen that was performed in the 1970s, where the scientists found that when this one gene was mutated, the larva actually started to form these sort of spiny needles. And so they called it hedgehog because they thought that these larvae actually resembled the hedgehog. And the first couple of hedgehog proteins that were identified in humans were given the names Desert and Indian. And then the next one was actually given the name Sonic for the Sega video game character. And probably, certainly the younger people in the audience will actually recognize Sonic Hedgehog. So this is a cute connection. But unfortunately, this specific gene, mutations in this gene when it comes to humans gives rise to pretty horrible things. And so, like I said, it's a morphogen that's involved in development. And we know that mutations in Sonic Hedgehog are associated with sort of major craniofacial deformation, as well as actually the cyclopia, where you have a single eye. So it's very sad. And there's been an effort for a Broad scientist to sort of think about this target. So Sonic Hedgehog, this is the crystal structure of the murine version of Sonic Hedgehog. Sonic Hedgehog is an extracellular growth factor, so it resides outside of cells, and it will bind to this receptor called patched. I won't go into all of the details about the signaling pathway, but when this binding interaction occurs, it sort of removes an inhibition step of the patched receptor to another protein called smoothened. And this will now then activate a signaling pathway which results in the transcription of many genes that are involved in either development or disease. This is called the GLE pathway. There are actually known agonists and antagonists of this pathway. There are both natural products and synthetic compounds that target some of the signaling enzymes in this pathway, as well as the smoothen protein, which is one of those druggable GPCR proteins. This has sort of been a major effort for researchers both in the academic community as well as the pharmaceutical and drug discovery community. We think, though, that as biologists, we're kind of interested in understanding what happens from a biological perspective when you inhibit this interaction, when you move upstream of smoothened. And so what we did, actually, it was a graduate student here at the Broad Institute named Ben Stanton. He took purified sonic hedgehog, he put it on the arrays, and he looked for binding interactions. And he actually identified a compound. We call it Robotnik. It's actually inspired by Dr. Robotnik, who in the video game antagonizes Sonic Hedgehog. So this compound is actually an antagonist of the hedgehog protein. And it binds. It's got a decent KD. It's low micromolar. The compound has an effect in cells, and this is an example where you see the compound different doses, and it can be activity can be rescued with one of those smoothened agonists that I showed on the last slide. We've worked with researchers at the MGH to actually understand the function of these compounds in primary keratinocytes, as well as synthetic models of human skin. And this project is now sort of what we call a flagship project here at the Broad, where we've got many, many different chemists and many different biologists who are trying to use tools like Robotnikinin to understand the role of Sonic Hedgehog in the development of different types of cancer, including pancreatic cancer. We've also already taken a compound like this compound here from micromolar affinities to picomolar affinities. And this is due in part to the nature of the types of compounds. And that goes back to the idea of assembling a transformative collection of small molecules. You'd like to have compounds in your collection that are easy to modify quickly. So you can make several analogs and understand what those analogs do. Sort of moving a little closer to what my group does. My group's been interested in the role of transcription factors, and so coming back to this original slide, we don't know what is responsible for organismal complexity, but when you ask, how many transcription factors are encoded by the genes in these different proteomes, you can see there's a bit of a bump here in humans. So. Transcription factors are proteins that come together in combinatorial assemblies to regulate the timing of gene expression. And so we think that the larger number of transcription factors and the fact they come together combinatorially may help give rise to organismal complexity. And this is not a unique hypothesis to my group. This is actually a very, very common hypothesis. Does everyone remember what a transcription factor is? No? All right, well, good. I'm glad I put this slide in there. So transcription factors are basically proteins that bind to DNA at very specific sequences, and they control the transcription of that sequence of that genetic information from DNA into RNA. And actually, we have a movie courtesy of Bob Tejan, who's the head of the Howard Hughes Medical Institute. So this is a very common transcription factor. John Wren talked about P53. P53 is going to come into a specific binding site adjacent to the gene that it's going to regulate. Here it comes. And it recruits an RNA polymerase, and that will come down, and then transcription will begin, and you will start to make mRNA. That later on, this mRNA will actually be translated into a protein. That protein can then go on in the cell to have affect function within the cell. So we think transcription factors are cool. They're also known to be important targets for cancer therapy. We're not going to go through all the details here, but Many of the signaling proteins, the usual suspects, will change patterns, change transcription patterns when they're mutated in cancer. My favorite types of transcription factors, the rapid-acting, latent cytoplasmic transcription factors, things that kind of hang out here and upon some sort of signaling response move into the nucleus and affect transcription. Many of those are aberrantly regulated in cancer. And you know, we think as chemists there may be selective sites of modulation with these different protein-protein interactions. But that's actually considered, protein-protein interactions are considered to be undruggable. So if you want to try and make small molecules that target transcription factors, this is still considered to be a complex task. So this is a model of, it's a DNA coil, and all of these little balls are actually different types of proteins. This is what comes together to actually affect transcription. And as I said, again, as a chemist, I know how to think about disrupting a chemical transformation that an enzyme catalyzes, but I have no idea how to design small molecules that will disrupt these different types of protein-protein interactions. So we think about screens again, and we've actually been thinking about our small molecule microarray screens. When I was a graduate student at Harvard, I ran a proof of concept experiment with transcription factors involving a yeast transcription factor that was originally discovered here at MIT. It's called the HAP complex. And what we wanted to do was pluck one of these proteins, HAP3, out of its normal complex from cells, purify it, stick it onto a microarray, see if we can find binders, and then ask the question, do those binders, when you reintroduce them in cells, affect the function of this transcription factor? So we identified these compounds that we called haptamides, and we put them into cells, and we found, using something called the reporter gene assay, that we could get a dose response. And at around somewhere between 50 and 100 micromolar haptamide, we could reach levels that resemble an otherwise isogenic deletion strain in yeast. So basically, this yeast is the same as the normal wild-type yeast, but it's just lacking our target. And we could reverse this. And I went into the lab and made a bunch of compounds, et cetera, and we were able to come up with better compounds. So that was really just a proof of concept that you could use this approach to target an undruggable class. Most of the people around here weren't so impressed about the fact that we were working in yeast, unfortunately. So if you're a yeast biologist, you should yell at people at the Broad Institute. But what we decided to do is to really scale this up and not just think about a single protein, but we came up with a collection of 100 different transcription factors. These are 100 different undruggable targets. And they were chosen for a variety of reasons. They have different types of functions. They have different types of structures. Some of these are what we call resident nuclear factors. So they stick around, and they're always living in the nucleus of a cell. Some of these are called nuclear hormone receptors. So this is an example of a transcription factor that's actually considered to be druggable because we know hormones, endogenous small molecules that bind to them. And then we have some proteins that are actually those latent cytoplasmic proteins. So they're fast-acting, rapid, ubiquitous proteins that will move quickly into the nucleus upon some sort of stimulus and then affect gene expression. We've got some members of the general transcriptional machinery. And then we've got some things that aren't probably proper transcription factors. They don't necessarily bind to DNA directly, but they interact with some of these other proteins. And then I'm actually just going to talk a little bit about these particular proteins here, the NF-kappa Bs. But before that, I just wanted to point out what we did was we had our proteins that were purified. They had a tag on them. We came in with that antibody again. We screened 20,000 different compounds against 100 proteins. We did that in triplicate. So if you think about how many spots you're looking at, it's 6.6 million. And what that converts into is 2.2 million theoretical protein small molecule interactions that we probed. Which is actually a pretty significant number. And like I said, many of these compounds come from different sources, including known drugs, natural products, and synthetic compounds. On the one sort of goal of our group is to take this massive data set and to try to understand what the rules are for targeting transcription factors. So we've annotated the compounds for certain types of descriptors. How many fluorines are in the molecule? How many rotatable bonds are there in the molecule? How many double bonds are there in the molecule? And we also annotated the proteins for certain types of biological descriptors, structural descriptors, functional descriptors. And what we want to do is use informatics to try and search for correlations between structure and binding. And maybe we'll see something that provides a clue about what types of compounds will be good for targeting transcription factors. All right, so NF-kappa B. So this is an example of one of those cool proteins that someone decided to give a really long name to. It's a nuclear factor kappa light chain enhancer of activated B cells. David Baltimore, who's a friend of the Bird Institute, used to be sort of across in the building next door, is one of the discoverers of this protein. It's one of the master immunomodulatory transcription factors. We like to call it NF-kappa B because we don't want to say this every time we talk about it. It's highly conserved. It's found in almost all cell types. It's one of those proteins that's involved in all sorts of cellular responses. It regulates many of the genes that control cell proliferation and survival. And when this particular protein becomes overactive, it's associated with many different types of cancers and other types of diseases. So this is sort of a schematic of what we call the canonical, it's like the major version of the NF-kappa B pathway. Hangs out in the nucleus, it's a latent cytoplasmic transcription factor. You get a signal and some sort of receptor, triggers the usual suspects to start doing their job, and then you form this heterodimer of one NF-kappa B and another NF-kappa B that translocates into the nucleus. It binds to other types of transcription factors, and then these genes that control cell proliferation and survival are turned on. There are about 700 compounds known to target different proteins, often the usual suspects in this pathway. But there are no compounds that directly target the transcription factor itself. So we thought that was a good challenge. And we put both P50 and P65 on our arrays, our magic glass slides. And we found a bunch of compounds. And these are some of the structures that bind. We, again, see things that look very, very different. We see some compounds that resemble each other. This is just a subset of the compounds. But we have a favorite one right now. It's also got a funny name. It's BRDK92758-126. Basically, this is how the Broad Institute names its compounds. This particular compound, we've now confirmed that this is a direct binder to both the P50 and P65 subunits. The compound binds with affinities in the mid-nanomolar range. And we use that tool called ChemBank I told you about, where you can see what a compound does in cellular assays. And we found that this particular compound, that it scored as a hit in 2 different types of cellular assays, and they both turn out to be related to the NF-kappa B pathway. So we thought this was kind of cool. We had some compounds that bind directly to NF-kappa B, and we know their cellular effects are actually related to NF-kappa B. So we thought we'd sniff a little further and use another tool that's been developed by the cancer program here at the Broad Institute called the connectivity map, CMAP. The goal of CMAP is to use a common language of genome-wide mRNA expression profiles to make connections between diseases, genes, and drugs. And so this is the URL. Again, it's one of those things where if you have a favorite compound, you can go in there and see what it does. But the idea is that basically you have a biological state of interest, and you generate a gene expression profile, and from that you come up with some sort of query signature. We won't go into the details, but if you go to the CMAP website, you can understand how this is done. So the folks in the cancer program here have actually built a pretty big reference database of different profiles. And those profiles include RNAi experiments. They include small molecule experiments. They include experiments with known drugs. And what you want to do is see if you can make a connection between your particular state of interest to this reference database and potentially make connections between different compounds, different states. So we thought our compound might be in connectivity map, and we were lucky that it was. It was actually profiled 4 different times and in 3 different cell types. And basically, to a little bit more about connectivity map, there's basically 6,000 different instances of profiles in the database. And there are 1,300 different compounds that have been profiled. So we generated many different types of query signatures involving different cell lines and different conditions, but no matter what type of signature we queried, each time the top 20 to 50 compounds that would cluster with our known compound here. They were things that were known to perturb the NF-kappa B pathway, so known pathway inhibitors, known pathway activators, or they were anti-inflammatory agents. So again, our compound that binds directly to NF-kappa B, when you ask what this does in cells, it's starting to smell like an NF-kappa B modulator. So then we decided to go in the lab, do some lab work. I teach a class at Harvard, in the Harvard biology department, where we have undergraduates, and we assign them with sort of different projects, different questions that we think are really relevant to the research in our own groups. And so I said, hey, we've got this compound. We want to know what does it activate or repress transcriptional activity in cells? Does it affect that translocation of the protein from the cytoplasm into the nucleus? Does it directly affect the ability of NF-kappa B to bind to DNA? So they went through and they went to the lab and they ran a bunch of experiments. And this is just some of the data shown here. This is a reporter gene assay. So with these cells, if we treat them with something called TNF-alpha, the pathway will be activated. You'll see lots of transcription. And then what we did was we took cells that had been stimulated with TNF-alpha, so they should have elevated transcription, and then we co-administered with different compounds that we knew bound to the transcription factor. And we saw repression of that TNF-alpha stimulated transcription. So that was good. It looked like our compound was actually an inhibitor of transcription. So then we asked, how does it affect that translocation process? So here you're looking at cells where the green stain is actually an antibody that's directly against NF-kappa B. So that's staining the NF-kappa B. We've got blue is staining the nucleus. So these cells have actually just been stimulated with TNF-alpha. And so you see green in both the nucleus, so an overlay of the blue and the green, and you see it in the cytoplasm as well. When we treat with this compound, you can actually see distinctly the blue nuclei. So the compound the protein is no longer moving into the nucleus. It's preventing that translocation. So right now, our group is trying to understand the mechanism of how this compound works. What does it do? Does it disrupt a specific protein-protein interaction? We've also identified compounds that are more potent. We've identified compounds, derivatives of the original compound, with nanomolar and actually picomolar EC50s in the assay. And with this large set of transcription factors, what we're doing now is collaborating with various experts on specific transcription factors within the community. We sort of feel by interacting with these people and moving these stories forward, we not only get to demonstrate the utility of our small molecule microarray approach, but we get to basically reverse the feeling that transcription factors are undruggable targets. In fact, we think transcription factors are imminently druggable targets. We're not only thinking about druggable targets, or undruggable targets. I wanted to just close on a class that is normally considered to be undruggable. So we'll revisit those histone deacetylases that I told you about at the beginning of the talk. These HDACs actually have a very conserved active site. These are the enzymes that catalyze some sort of chemistry. And there are known clinical agents that bind to these enzyme active sites. These active sites are very highly conserved, and it's been a challenge, both in academia and in the pharmaceutical industry, to identify compounds that will distinguish between HDACs. So there are actually 11 zinc-dependent HDACs, and there are compounds that will bind to all of them. So what Jason Fuller, who is in the audience, did was he came up with a way where he would take the proteins and he would pre-incubate them with the known clinical agent, vorinostat or SAHA. And then he would apply that protein with the agent onto the array. And the idea is that he would look for proteins that might bind outside of the active site. And the idea for HDACs is that most of the differences in these proteins are outside of the conserved enzyme active site. And so Jason did this, and he found a bunch of compounds. This is, again, sort of the idea. Different compounds might bind to these different binding clefts outside of the conserved active site. And we were able to identify compounds that specifically target different HDACs, including HDAC1. And sometimes we found things that weren't so specific. So we're now actually using this approach, not just on HDAC1 or HDAC8, but we're trying to look for binding signatures across the entire class of histone deacylases. We want to find things that bind to one HDAC or two HDACs or all HDACs, because the idea now is that these individual proteins actually play very different roles in biology. HDAC5 might be very important for psychiatric diseases. Another protein, HDAC3, might be very important for ovarian diseases. And we'd like to use small molecules that are specific to an individual isoform of these HDACs to understand the underlying biology. So yeah, this is an example of how we're using the same technology to find novel chemical matter against what's traditionally been considered a druggable class. And so to close, I guess the statement I would make is that undruggable proteins are amenable to small molecule science. Are undruggable proteins undruggable? I think we're going to have to let the pharmaceutical industry tell us if that's the case. But by developing novel screening collections, developing novel screening technologies, we can find gold standard probes of these undruggable proteins. And if we do that, we may actually lower the barrier for commercial groups to actually go in and start looking at the same targets. And that may be the way that we actually go from a paltry 400 plus druggable targets to maybe tens of thousands. So with that, the most important slide is this slide, which thanks the very, very large group of people that I've had the pleasure to work with during my time at the Broad Institute. As I said before, it's chemists, it's biologists, it's people who work in informatics, and you know, a couple of names that I'll sort of give a shout out to. Olivia McPherson is actually the person who's really worked on a lot of the small molecule microchemistry. Katie Dowd is someone who's worked on both the NF-kappa B and the sonic hedgehog project. Where's Jason? Jason Fuller has actually been the one who's been running most of the histone deacetylase project. And Rakib Busanelli, who's actually a relatively new person here at the Broad Institute, has really been pushing hard on the NF-kappa B project. We also are lucky enough to have lots of collaborations with the different platforms here at the Broad Institute, the chemical biology, novel therapeutics, cancer, and the proteomics platform as well. And so with these people, we're actually able to think about tackling the undruggable. With that, I'd be happy to take any questions. If there's any time. Yeah.
You mentioned so many screening technologies. Just wondering which one you think is better, or it would be better to use to identify molecules that help induce ideal cells to certain cell lineages.
That's a great question, and so what I would say is actually you have to take many different approaches to that particular problem. So we can actually identify compounds that bind to specific targets that may be relevant to reprogramming. And actually, there are projects underway here at the Broad where we might take certain transcription factors or growth factors that are an important part of that process. But there are other people here who are thinking about just running cellular assays as well. And we're often looking at the intersection of the in vitro assays as well as the state switching type assays. But there is a strong effort in reprogramming here at the Broad Institute, not focused on a single technology. Yep. Any other questions? Another one, go for it.
Some of the undruggable proteins,
yeah.
You mentioned that some of them are transcribed from majority of genes. I mean, how is it possible to have one protein that has been transcribed from majority?
Oh, no, sorry. That's not what I actually meant. So the majority of proteins that are possible are undruggable. So we're only looking at something like 400, 300, and that's debatable, proteins which are considered druggable because they've already been drugged. But the majority of the majority of potential proteins are considered to be undruggable. Yeah. Okay. Yeah.
Curious how the pharmaceutical industry views the work that you do.
How the pharmaceutical industry views the work that I do, specifically the small molecule microarrays, or just generally what's going on at the Broad Institute? You know, We actually have interactions with different pharmaceutical groups. So you could imagine that if one of these particular compounds is doing something really cool to a target that they have an interest in, but just conventional screening approaches haven't allowed them to access that space, that they might have an interest in potentially moving that compound forward. I can tell you here I have been contacted by too many people from industry and academia that want to put their favorite proteins on the arrays, and it's just not something that we could do here. There's actually been too much demand for us to think about what different pharmaceutical groups want to do. I mean, to be honest, it seems like every pharma that you can throw a stone at in this local area has a list of 100 targets or more that they'd love to access, but it's either too expensive to execute 100 different campaigns. It's too much assay development involved. And if there's sort of a cheaper, just kind of, put on the boots and jump in the mud approach, they're often willing to sit down and talk with you about it. So, the other sort of aspects of screening, I mean, I think it's sort of, people are interested in cell-based screens as well. But as I mentioned, there's this challenge of identifying the targets. And at the Broad Institute, we have an answer to that, and a number of the pharmaceutical groups around here are developing their answers to that problem too. Yeah.
Is there a computer science effort that are trying to predict the interactions before they actually occur, or trying to explain the interactions afterwards?
So that's a great question. So is there a computational effort to try and understand in advance the types of interactions that might occur, or sort of understand them retrospectively? So I wish the answer was yes to the first, but right now it's really not so much the case. We're not running those types of predictive experiments, at least in chemical biology. We're kind of hoping that these types of data sets generated using small molecule microarrays and other screening types will allow us to run those types of predictive models in the future. On a case-by-case basis, there may be specific targets that a group is using modeling approaches to try and do virtual screening. But at the Broad Institute, there's no significant virtual screening effort. There are such efforts at places like UCSF, for example, but not at the Broad.